All right. Hello. Welcome back to our last session. My name is Kevin Koss. I'm a postdoc at the Danforth Center, and I'm excited to be your moderator for this last session. So this last session will cover techniques over spatial omics and plant biology. And our first speaker will be Bertrand Yeung from Curio Bioscences. And before Bertrand starts, he has to leave for a flight shortly after this presentation, so he will not be able to participate in the panel discussion. So to kind of help him get some Q&A and get some discussion, immediately after his presentation is over, we'll open up a 5-minute Q&A discussion. So if you have questions for Bertrand, please put them in the chat so that way he can answer them before he leaves. So with that, Bertrand, I'll turn it over to you.
Okay, great. Thanks for the introduction. Yeah, so I apologize for that. I have to go to a conference today, but let me share my screen. All right. Okay, can you see it? Oh, good. Slideshow. Okay, good. Okay, awesome. So yeah, so thank you guys for having me. I'm using my Plant Cell Atlas cup. I don't know if you can see it, but it's it's here with me. But anyway, so our company is Curio Biosciences. I'm the director of business development. What we are is a company based in Palo Alto, California, and we are commercializing scSeq. And so I'm just going to skip straight to the technology itself. Just like in the original scSeq paper, the resolution is 10 microns. Tile area size is 3 by 3 millimeters. It is whole transcriptome, so it is using a poly-T capture. For the area of 3 by 3, if the tissue is covering the whole tile, we do recommend a sequencing depth of 200 million reads. Of course, if your tissue is more highly active, you probably will need to sequence more. Currently, there's no specialized hardware that's needed for this assay. It's very easy to use. It's compatible with fresh frozen tissue. Throughput is really user-defined, depends on how familiar you are with single-cell assays or even spatial assays. As long as you have a good amount of experience, it should be very straightforward for you. But even if not, we have some new users who haven't really done genomics before, and they're still able to run this. Workflow is total of 8 hours, about 2 to 3 hours of actual hands-on time. And we do have a dedicated primary analysis bioinformatics pipeline just to be able to convert the FASTQs to the matrix files that you need for secondary analysis. This is the actual kits. So on the left, you'll see a few boxes and then a pouch with the actual tiles in them. There's one box that goes into -20, one at room temperature, and then the slides themselves go in the 4 degrees. Shelf life is about a year from date of manufacture. And on the right, you'll see the actual tile, where the white spot in the middle is where all the beads are. These are 10 micron beads, so there's about 80,000 of these beads in a small 3 by 3 millimeter area. And this is mounted on a blue piece of tape within a plastic cartridge that's about the size of a normal glass slide. Okay, so now onto the more interesting stuff. I'm just going to show you some data. This is the workflow. Sorry, I'll go through the workflow, but the workflow is you mount your tissue on the slide. It'll go through cDNA synthesis with RT and tissue digestion in an Eppendorf tube. So you don't need to worry about losing anything in your sample. And the rest of the protocol is just an Eppendorf tubes and PCR tubes and very standard library prep. So data. Okay, so now I am showing you animal data first, obviously, because that's what we have internally. This is just an example of mouse hippocampus. As you can see, this is just total UMI counts in log 10 scale. So depending on where you are within the tissue, you're getting several thousand UMIs per bead. Obviously, there'll be areas that are much lower, where there's lower transcriptional activity or where there are no cell bodies. If you do go into secondary analysis, you can clearly see structure within the tissue, depending on the genes you're looking at. And more recently, last month, from Joe Ecker's lab, they published a paper in bioRxiv where they were using our kits, looking at Arabidopsis seeds, different stages of development. These are just the generic graphs. You'll see multiple seeds are mounted within the tissue block and then sectioned all together and then mounted on the tile. And so you're getting pretty good UMI capture across the board. And then what they mentioned was that the annotation of cell types requires prior knowledge of validated cell types, cell type specific markers. And this isn't always available for tissues where there isn't much data, right? So these ground truths aren't always there to define what these cell types are. And so what they used was Seeker, or a spatial platform, to validate the several de novo identified clusters. So basically clusters that they identified, but they weren't quite sure what type of cells they were. And so I'm not a plant biologist, so you guys probably understand this way more than I do, but this, on the left, you can see the UMAP clustering of the different cell types, as well as the actual spatial plots of where these clusters are landing. And on the right are just zoomed in versions of these different images on the left. So you can see different structures again within the tissue. I thought it was pretty cool. You could see the seed coat surrounding the seed, which makes sense. And also, so we are doing larger tiles now. We have a 10 by 10 millimeter tile that's in early access. Same high resolution as a smaller one, just more beads. So there's probably about 1 million beads on this tile. Good capture efficiencies, very similar data. A lot of genes per bead, a lot of UMIs per bead, and then you can see it has really good clustering for the tissue, as well as the spatial reconstruction of the clusters. And then, secondary analysis looking at structures within the tissue as well. One thing I'll mention, for at least for the data set from Joe Ecker's lab, they didn't need to adjust the protocol at all. It just happened to work right out of the box. We had been anticipating that we would need to adjust it because of the cell wall, trying to digest it completely and get it to solution, but apparently that wasn't the case, at least for this sample type. But I know there are other groups within the plant cell atlas that are looking at the protocol, seeing if any adjustments need to be made for the enzyme composition. And that's it. I'm gonna try to speed through that as quickly as possible. So if there are any questions, I'll take them now.
Awesome.
Yeah, go ahead.
Yeah, no, it's all good. Yeah, thank you for that, Bertrand. So yeah, as a reminder, if you have any questions for Bertrand, please put them in the chat and I'll be able to go through them. Yeah, I guess I'll start just to break the ice a little bit. Sure. So I guess what is kind of needed from like a user standpoint. So it's a kind of like we just prepare the samples and then we put them on the slide and then we ship them back to you, or do we have to process the data ourselves and then you kind of have softwares?
Right. So this is just a kit that we sell. We send the whole kit to the customer. They do all the tissue mounting on the tile, all the library prep, sequencing. We do have the primary analysis pipeline that we can have you guys install locally on your local servers. And then once you process it through, then usually it's up to the customer what secondary pipeline they want to use. We do have a Seurat output file, so if people are using Seurat, they can use that, but also just a generic matrix file for whatever secondary pipeline you want to use. We are working on a cloud-based version of the primary analysis pipeline. So for people that aren't as familiar with informatics, they'll be able to just upload the FASTQs to the cloud in that program and then process it there. They'll just get the HTML report and all the other files as well.
Awesome. Yeah. Okay, so we got one question here. So a question was asked, what is the spatial resolution?
Yeah, the spatial resolution is 10 microns. The beads are right up next to each other, so the pitch is technically 10 microns. So there shouldn't be really any space in between the beads.
Cool. We have another question where it was asked, in a plot with the genes or UMIs per bead, it seemed that there were signal outside the seeds, but in the clustering results, there was no signal outside the seeds. So
Right.
we're able to filter out.
Yeah.
info.
Yeah. So what we found that when you mount tissue in OCT, sometimes if you don't clean it completely, there may be RNA that are either leaking out while you're freezing it into the OCT or diffusing. So usually we recommend, washing the tissue or whatever you're doing very well, and then dry it, and then as much as you can. I mean, don't tap it with anything that's contaminated, but try to get it as properly mounted as possible in the OCT without any other solution in there. So usually what we do is just bioinformatically we're able to take those out or filter those out, because they generally have lower read counts per bead, like much lower, maybe in the 10s or 50s or something like that. And it's usually fine. But it's just goes to show it's extremely sensitive, so it will pick up anything that's there, that's floating around. But yeah, it's usually nothing from the tissue itself when you're sectioning.
Gotcha. Yeah, and that's actually kind of leads like to another question that is in the chat. It's like, so since you have these beads like really close together from like, they can catch probably like 2 adjacent cells, is there any issue with bioinformatics or deconvolution of the data?
Yeah, so deconvolution is something that you guys use in other platforms as well, and you can use here. Internally, we use RCTD, which is used for integrating single cell data into the spatial data if you really want to deconvolve the data. So you can tell which beads are 1 cell, 2 cells, or you can do it by fraction if they're multiple, but usually they're on the order of one or 2 cells. Of course, for plants, there are much larger cell types as well, and so it could be subcellular. But we're working on. I mean, there are some platforms or bioinformatics pipelines out there that can overlay an adjacent section with H&E staining or whatever other staining you're doing as well, to figure out if you need to define those cell boundaries. But at least in animal tissue, for 10 microns, we're kind of smack in the middle between, like, in single cell territory. So there's some cells that are smaller, some that are larger. So you will expect that some beads will have more than 1 cell in it.
Awesome.
Yeah.
Cool. And I guess with that, I would like to thank you again for your talk, and have a safe travels. Okay. And if you guys have any more questions for Bertrand that may come up, feel free to reach out to him. I'm sure he'll be happy to answer you over email. Yeah, just email me, or you can send it to somebody and then they can email me all the questions. I can answer them later. Okay. Absolutely. Thank you. Thank you. Yeah.